Hello, I'm going to tell you about my PSE project. It is about the evolution of some ribosomes. The central topic will be the origin of life. One of the theories that helps to address this topic is the RNA World hypothesis, in which self-replicating RNA molecules proliferated before DNA or proteins. For example, in the primordial soup at the origin of life on Earth, they could have been nucleotides that joined randomly. Some of these small sequences, known as oligomers, could grow by a temple-based ligation process, causing the sequences to replicate. In this way, we could have arrived at much complex sequences of RNA that could have a catalytic activity. That's ribosomes. These ribosomes could be the key in the development of the first cell. Temple-based ligation is ligation held by a complementary sequence on which the attachment of small fragments to a template can help the replication of the main sequence. But it's not only important the fact that they could replicate, but how they did it. Imagine in the primitive Earth in an ocean. To keep the sequences close to each other and prevent losing information by diffusion, their growth had to be exponential, but with no reliability so that the sequence was contained, but at the same time that it could evolve in the primitive environment of the search of the first cell. This is how we came interested into ribosomes from bacteria and viruses. One, Azaroachus, can self-assemble, and the other one cannot. But in both cases, it has been shown that they could give ligation reactions. It is important because the main goal is finding a ribosome capable of self-replication that has evolutionary potential. Thus, my project is based on observing sub-modifications of the sequence of the ribosome where the substrate binds affect the catalytic activity of the ribosome. Then, another objective is the optimization of the binding of the ribosome with respect of the environmental conditions, such as buffers and metal concentrations. For this, DNA sequences of ribosomes and some substrates were ordered, and with in vitro transcription, extraction by phenylchlorophore method, and column purification of the RNA sequences, the final sequences were obtained. Now, for the catalytic assay, two systems are used. One, in which the appearance of a band in electrophoresis gel is evaluated, because it has an increase in molecular weight after ligation, and the other system in which the increase is in the concentration of the band at a given molecular weight. For both cases, the mechanism of ligation is unknown, but it is known that these G nucleotides are essential, so they cannot be ligation without them. Here we observe the results of the first analysis. For this case, we observe bands at the expected molecular weights, but to compare them, it is necessary to sequence them, something that I couldn't do throughout my project. For the second case, there is a case in which product formation could not be seen, but is not the main one, so we would have to repeat the experiment with different sequences and subsequent sequences of the products obtained. It is important to note that it could not have been secondary structure because the gel conditions prevented. However, I had problems with the production of Azaroachus and I couldn't continue working with it. So I decided to compare with the ribosome from which the sequences changes I made in Azaroachus had been inspired, Sunway. While it seems in the beginning that Sunway produces more easily the bound product, at the end it is observed more dark bands for Azaroachus, so I decided to quantify it, and indeed Azaroachus produces under the same amount of products of substrate a higher amount of product. But in the beginning it was promising, so I decided to optimize the experimental conditions of Sunway to compete with Azaroachus. This optimization is important because the more product you have, the study of ligation mechanisms becomes easier, and therefore the finding of a ribosome that is self-replicating and able to evolve. So I used 3 media, water, and 2 traditional biochemistry buffers on the magnesium concentration between 0 and 100 millimolar. We see there is no reaction without the metal, and also the differences between bands are subtle, so we quantify again. We look at the results and see 2 important things. One is that the optimal medium, based on the final amount of substrate after 24 hours of reaction, is water, with a concentration of magnesium between 75 and 100 millimolar. Also, a rapid initial growth is observed with a resting phase. It could be thought that this kinetics comes from the fact that it is an exponential growth, but further validation is needed. So finally, we arrive to the conclusions. We see that modified versions of Azaroachus could catalyze ligation reactions even better than more traditional ribosomes, such as Sunway, though the sequencing of the products is needed because we need to understand more the mechanism on how it works. And for Sunway, in the end, even at the optimal conditions of 1700 and 100 millimolar magnesium chloride in water, it could not arrive to the same amount of product as for Azaroachus.